Good morning, everyone. Welcome to Takara Bio's Smarter NGS webinar series. Our topic for today is going to be a smarter solution to single-cell human TCR repertoire profiling. We have two speakers for today, Dr. Sarah Taylor. She's the lead R&D scientist for all our TCR immune profiling products. And today, Sarah is going to show us her recent developments for the single-cell TCR kit. And the second speaker is myself. I'm the marketing specialist in the NGS group. So moving along with the agenda, we're going to briefly review why we study the TCR repertoire, show some application data on the smarter human TCR profiling kit. We will then review briefly why perform single-cell TCR profiling, show you some background on the smarter human TCR profiling kit, and then Sarah is going to end with some application data using the single-cell TCR kit. So shown here in this cartoon is a B-cell and T-cell repertoire. So in a functional adaptive immune system, what you have is a very diverse repertoire of T cells and B cells. And upon being presented with a MHC class molecule, the TCR receptor recognizes a foreign antigen and the T cells can mount a cell-based immune response. The T cell receptor shown here is a heterodimer primarily of the alpha and beta chains. And in a minority of population, you also have gamma and delta chains. So as highlighted here, this is I'm going to review how this diversity is occurring in the germline. So shown here is a segment of the germline TCR beta DNA. It has multiple gene segments, the variable and the joint genes, as well as a constant region. So what happens, and shown here is the TCR beta DNA, what happens in the germline is these gene segments undergo a site-specific recombination to give rise to this rearranged segment. In addition, there are additional mutations that happen at the junction region, giving rise to the final gene segment shown here. Then there is splicing and transcription and splicing occurring, which gives rise to the final product shown here, the VDJ in case of TCR beta and VJ in case of TCR alpha, followed by the constant region sequence. The functional receptor, again shown here, is a heterodimer of alpha and beta. What's also important to note is that in this junctional region, there are hypervariable loops called the CDR1, 2, and 3, and the CDR3 sequence is very important it interacts directly with the antigen and is responsible for antigen binding. Therefore, sequencing the full-length CDR3 is critical for understanding antigen specificity of the T-cell receptors. So moving along, recently there have been 3 methods to study TCR diversity by NGS. And this is a picture from a review article published by Rosati et al., where the 3 main approaches are presented here. Multiplex PCR, target enrichment, and 5' raise combined with template switching cDNA synthesis. I'm going to briefly review each method and the benefit of doing multiplex PCR versus target enrichment, and try to highlight why we have picked the 5' raise method for our method of choice in our TCR profiling kits. So as mentioned, in the multiplex PCR, you can start with RNA or DNA template, and what you do is you design gene-specific primers to the variable and constant regions, and you amplify them, and then in a subsequent round, you incorporate Illumina adapters for sequencing. In this approach, you only can amplify a portion of the variable region, and because it's a PCR-based approach, there is a possibility for amplification biases, as well as since you have primers to a set series of genes, you are unable to detect new V alleles due to the fixed primers used. In the other approach, the target enrichment, you can again use genomic DNA or RNA as template, and while this approach requires fewer PCR cycles, therefore there is less bias, you may still have specificity issues of amplification or enrichment due to differences in hybridization, as well as other issues, specificity issues. Now, the third method represented here is the race-based, 5' race-based method. And what you do here is you do oligo-DT or random priming of the RNA. And once you reach the 5' end of the RNA template, you then do template switching and incorporate Illumina adapters or sequences of choice. In this particular approach, since you use mRNA as template, you get a better representation of what's been expressed in the cells, and because there is no PCR involved, you avoid amplification biases and get fewer unexpressed or non-functional sequences. So at Takara Bio, we have used the template switching technology and incorporated it in our immune repertoire kit. So shown here is a portfolio, our entire portfolio of all the immune repertoire profiling products. As you can see here, we have kits for profiling both from human and mouse samples. In the past, we've launched a bulk TCR kit for human as well as mouse and a BCR kit. And most recently, we've added to our portfolio the single cell TCR AB or alpha beta profiling kit. Now, all of these kits with all of these kits, you can obtain complete VDJ sequence information where you get full-length CDR3 and can for alpha beta for the TCR and heavy kappa lambda for BCR. You can there is a flexibility in the input range. You can start with as little as 10 nanogram to 3 microgram of RNA or purified cells, and you obtain Illumina sequencing ready libraries without doing ligation. So moving along to explain a little bit of the smart cDNA synthesis mechanism, which is the basis for all of our kits. So what's shown here in cartoon is blue is the mRNA template, and in green is the smart or the oligo that is going to use for the cDNA synthesis. So what you do here is first-strand synthesis using MMLV-RT enzyme, which performs the cDNA synthesis, and when it gets to the 5' end of the RNA, it has the ability to add in non-templated nucleotides, shown here by these X's. What you do then is you add in this template switching oligo, which has sequence complementarity and can hybridize to these non-templated nucleotides. And then this enzyme is going to use this template switching oligo as a template to extend and add in more bases of your choice to the cDNA product. You can then add in primers and amplify to subsequently generate double-stranded full-length cDNAs. With this approach, you can obtain full-length cDNAs and incorporate sequences of your choice. For example, we will incorporate Illumina adapters, and because you are incorporating the 5' end, you don't need to have the full-length sequence of the variable region. I would then like to review briefly the strategy that has been used in the bulk human TCR kit. In this kit, what we do is we start with DT priming, and again, you perform a first-strand synthesis, add in non-templated nucleotides, and in this case, we add in the SmartSeq V4 oligonucleotide as a template switching oligo, which is the oligo that's been used in several of our single-cell kits. And after you obtain the cDNA and do extension by RT, in this case, you have 2 rounds of semi-nested PCR that we have incorporated. So in the first PCR, what you do is you add in a smart primer where it has complementarity with the smart sequence and the Illumina read primer too. In the reverse primer, you have incorporated part of the constant region of the human or mouse for the TCR alpha or beta locus. By doing that, what you end up with is a cDNA which now has Illumina sequences as well as part of the constant region. In PCR2, you then incorporate more of the Illumina indexes and forward reverse primers. So by doing this, what you end up as a final product is a full-length sequence of the variable region, part of the constant region, and Illumina adapters incorporated, giving you a sequencing-ready library. In the next slide, I would like to review some data we obtained with a collaborator using the Smarter Human TCR AB profiling kit. We collaborated with a group who was interested in examining repertoire diversity in patients with T-cell lymphomas. PBMCs were isolated and RNA was extracted using and then the smarter TCR kit was used to generate libraries. These libraries were sequenced on an Illumina MySeq with paired 300 reads, base pair reads, and analysis was performed using the Mixer software. To show you some data from 2 patients here, so what you have here are these chord diagrams which represent the clonotypes of the J and V genes expressed in alpha and beta. Each of these strings represent the pairing of the VJ, and the width of the line represents the frequency of expression of these clonotypes. What's shown here again is for the TCR alpha locus, you have a diverse range of clonotypes expressed with just this one clonotype, it's hard to see, being expanded in this particular sample. For the beta locus, you see the same thing, and you notice that there are a few of the VJ pairs that have been expanded. Patient 2 here with prolymphocytic leukemia, you see that for the alpha locus, there is an expansion of this one particular clonotype, both for alpha and beta. So to summarize this part, with this smarter human TCR AB profiling kit, you are able to get information on clonotype diversity as well as identify changes in the clonotype for TCR alpha and beta pairs. So now I would like to introduce why we decided to develop the single cell TCR kit. So in a cartoon shown here, what you have is a bulk input where there is a diverse population of cells. And if you do TCR alpha beta profiling, what you end up with are these range of clonotypes, where in the scenario you have 45% of this particular alpha and beta genes being expressed. Now, if you use this information to try and determine pairing, what you can determine is based on this percentage of expression, you can assign this particular AB pairing to the cell type, but what you're left with then is 4 combinations of alpha and beta expression that can belong to these cells. So using this approach, it is not possible to assign alpha beta pairing to individual cells. It has been shown that alpha beta pairing is critical to understand antigen specificity and recently has been used to design therapeutic antibodies. Furthermore, information on the lineage and history of cells are obtained by single cell TCR profiling, which could be important for studying development and diseases of the T cells. Furthermore, single cell TCR profiling allows one to understand the heterogeneity in the population of T cells during an immune response. Now I would like to move on quick to review the single cell profiling kit, showing you the workflow of the single cell TCR profiling kit here. So just as a reminder, this is the workflow of our bulk kit, and we have adapted the similar approach to optimize the results for the single cell TCR kit. So just shown here again, we're using a smart cDNA synthesis approach here, where we start with the DT sequence, and we are going to do first-strand cDNA synthesis and template switching. Now, as notice here that this template switching oligo has an indexed oligonucleotide shown here in pink, and what we include in this kit are 8 different indexes. The purpose of this is that when we start with a 96 well plate and we're going to pool later on to have 12 libraries, these indexes can then act as cell barcodes, which will then be used to assign specific reads to specific cells. Now, once we have done the first strand synthesis and extension by RT, we're then going to move into our gene-specific priming step. But before we do that, notice here there is an additional step shown of cDNA amplification with the cDNA primer. And the reason we incorporated that step here is because during the development of the kit, we noticed that from this single cell cDNA synthesis, we did not obtain sufficient yield for us to move into the gene-specific priming, TCR amplification. So therefore, we have this additional step where we then get enough cDNA to move into the next step. Now, what we do here is after the cDNA amplification, we do a pooling, which I will explain in the next slide, where we take the 96 samples and pool them into 12 to move into the TCR-specific amplification. So just let me briefly review what happens here. Is again, like the other kit, we have incorporated on the 5' end is a TCR primer 1 where you have Illumina read primer 2, the smart sequence. And on the reverse primer, you have the constant region from the alpha or beta. By this PCR, what you end up with is a double-stranded product where you have incorporated part of the read primer 2 as well as the constant region sequence. Then you do an additional round of nested PCR where now you have incorporated the Illumina indexes and have complementarity to this read primer 2 as well as part of the reverse index. So what you eventually end up with again is a full-length library with Illumina indexes and barcodes. Now I'd like to highlight again just for users that have used both the bulk kit and would like to try our single cell kit, we do not recommend mixing of components simply because in the template switching oligo we have a very different design and we included a cell barcode and we've added in additional primers in this step to improve our performance. Therefore, the components of this kit should be separately purchased for your application. So I mentioned in the previous slide that we do a pooling to help with the ease of use. So now I'd like to review the experimental setup on how the pooling strategy works. So in our user manual, this is a figure from the user manual. We recommend setting up negative and positive controls for your experiment to set thresholds for clonotype calling. So for negative control, here is a no template control. And for positive control, we use 5 picograms of jerkat RNA. So you have a negative and positive control plate. And what you do is you sort cells into a 96-well plate, and you could proceed to cDNA synthesis or freeze those cells and use later. So after you've transferred cells into the plate, the way the experimental setup works is you make 8 different master mixes for the full plate with the RT mix and the smart index oligos, and dispense each mix horizontally such that each row of cells have their own index oligo. You then perform 96 individual cDNA synthesis reactions, and then you're going to do the pooling. So this is how the pooling works. Shown here in this arrow, essentially you take all the cells or all the cDNAs from row 1 into one pool shown here. So you're pooling vertically. What that does is all these 8 individual cell libraries are now going to have different indexed oligos, which is then going to be used for demultiplexing later on. So after you've generated these 12 libraries and done the cleanup, you then do the TCR-specific amplification and library generation. With that, I'd like to turn this presentation to Sarah for her to present the performance data and application data that have been generated using this kit.
Thank you, Ankita. So I'm going to start by talking about demultiplexing. Because we use the 8 different cell-based cell barcodes that we pool, after you've performed sequencing, you need to demultiplex those based on those barcodes in order to perform data analysis. So we have some demultiplexing software that's available on the Takara Bio USA website that you can download. And using this software, you can select how many errors you'd like to allow in the barcode when performing the demultiplexing. So here I did an experiment where I took the same data set and demultiplex, allowing for 0 errors or 1 error in the barcode. And as you can see, when 1 error is allowed, you get slightly more reads mapping to each of the barcodes, and that increases even across all the barcodes, showing that there's no bias. And you get fewer reads mapping to the bad barcode, which is the unassigned reads. So you can use either 0 or 1 errors, and the number of reads, or the percentage of reads that you get that are unassigned is reduced accordingly. But we wanted to check that whether we're using 0 or 1 errors, the data looks the same. And this graph on the right here shows you each of the different barcodes with 0 or 1 errors allowed, and then the clonotype calls that are made. So for example, A0 is barcode A, 0 errors, And you see here there's about 45% of TCR alpha and 55% of TCR beta. When you look at barcode A with one error around, you see the same data. So that gives us confidence that either 0 or 1 errors can be used for demultiplexing. In the data I'm going to show in this presentation, I've demultiplexed using 1 error, just because that allows slightly more reads to map to the indexes and fewer to go to the bad barcode. So here I'm looking at whether the specific sequences in the barcode introduce any bias, and it's very important that they don't. So we wanted just to assess and ensure that that wasn't the case. So here I took 7 replicate pools, and each pool contained 8 times 5 picograms of JERCAT RNA. I'm using RNA here because we know then that there's no biological variation. Any variation we see is purely technical. So we took 7 pools that were run across 3 separate experiments that were all run on different days. And what you can see is when we average these data, and the bars here are the standard deviation, you see across the board there isn't really any difference between the barcodes. There's certainly none that stand out as being particularly high or low. And so we don't believe that there's any bias caused by the different sequences. And as I mentioned, I demultiplexed these allowing for 1 error, so our bad barcode pool is around 3% of the total reads. So now I'm going to show you some data we generate in-house to assess the performance of the workflow. And as Ankita explained, this is how we lay out the plates. And for this experiment, I took JERCAT cells and CCRF-CEM cells, 2 monoclonal T-cell lines, and I mixed them at a 1-to-1 ratio. They were loaded into the plate, including the controls, as we describe in the user manual. And then to validate the libraries at the end of the workflow, we run the bioanalyzer. And so what we would expect to see in the negative and the positive controls are something that looks like this. Negative control should be completely flat, so there's no yield, there's no template, so we shouldn't be seeing any amplification. In the positive control, we see this nice dual peak, sort of around 700 to 900, 1,000 base pairs. And this corresponds to the beta and the alpha chain of the TCR. And because this is JERCAT, it's very well defined. So then I pulled out 3 pools from this plate where I have these mixed cells. And I just want to show you these because I want to sort of highlight the variation that can be seen in the bioanalyzer profiles. In pool 9, we just see 1 peak, and that represents dominance of either TCR alpha or beta in that pool. Pool 10, you see the nice dual peak, so it sort of represents that alpha and beta are present at even amounts. And pool 11 is slightly skewed one way. Once we're happy with our bioanalyzer traces, we can move into sequencing. So we quantify our yield based on the bioanalyzer trace, and then sequence using a MySeq with paired 300 base pair reads. So after demultiplexing, allowing for 1 error, I use Mixer to call the clonotypes for these cells. And in order to have confidence in our clonotype calls, we need to set a threshold, so we won't be including any background reads, essentially. So to do that, I take the negative control wells and take the mean number of reads that map to the top 2 clonotypes in those wells. Now, even though these wells are negative, you do get a few reads going to them, and it's usually a few hundred compared to a few thousand going to the cell wells. And that's what we would expect for single cell sequencing. So we take the mean and then also take 3 standard deviations from the mean, because that means 99.85. If you look at this one-sided percent of the data, the negative data should fall within that range. And then we set that as our threshold. So if you look at the data on the right, here's a data set prior to the threshold being applied. All the blue dots represent each well, and the red dots represent the negative wells. When we take the mean and the 3 standard deviations of those negative wells and set the threshold, which in this case was a bit over 200 reads, we can then throw out all of the wells that fall below that and take all of the wells above it forward for further analysis. So going back to the experiment with the mixed cell population, sort of bringing that back here, we'll just go over that again. So we mix the cells, we go through our standard workflow as I've just described, and then based on the clonotypes that are called, we can assign a cell type because we know the specific clonotype that we are expecting to see for the JERCAT cells or the CCRF cells. And when we assign the clonotype across the plate, here's a sort of map of what the plate would look like. And when we count them up, you see that we do get approximately even counts of JERCAT and CCRF cells. So we get true representation of what we put in. We then look at the alpha-beta pairing in each well. And here's outlined, we get 14 wells where we only see the alpha chain, 41 wells where we only see the beta chain, and 34 wells where we see the pair. The omitted wells here are those that have fallen below the read threshold. So a summary of the pairing rate for the two different cell types. In the JERCAT cells, we see 56% pairing. In the CCRF cells, it's much lower, it's 24%, but that's because TCR alpha is expressed at a very low level in this cell line, so it's only detected sometimes. But as I'll show you in the data, what we generated through some collaborations, our overall pairing rate is in the 50 to 70% range. So here's the application data. I'm going to talk about two collaborations. One, we worked with some collaborators to look at PBMCs, and in another one, we looked at CD4 positive T cells. So the first collaboration I'll talk about will be the one where we performed the protocol on PBMCs, and this was performed with our colleagues at Takara Bio in Japan. The PBMCs were treated with OKT3, which induces proliferation of the T cells, and then the cells were sorted into 96 well plates. Now the group processed 2 plates and included controls as shown below. So not only did they include the controls that we highlight in the user manual, but they also included JERCAT cell controls. So when we analyze the control wells, as I described previously, we take the negative control wells and we set a read count threshold for confident clonotype calling. And then when we look at the positive control wells in this case, if we look at the JERCAT wells, we see the alpha-beta pair in all 8 of the wells across the 2 plates. When we look at the JERCAT cell wells, there are 12 cells included in the analysis. So I should mention that from the 2 plates, 3 pools actually weren't sequenced because they had low yields. So that meant overall 12 JERCAT cells were included in the final analysis across 2 plates. One of them had clonotype read counts below the threshold. So that leaves us with 11 cells. In 9 of them we see the alpha-beta pair, and in 2 we just see TCR beta. When we analysed the PBMC cell wells, we looked at 138 cells across the 2 plates. 44 of these fell below the read count threshold. So then of the 93 cells analysed, we looked at what TCR alpha-beta pairing we got. And we see it in 69 of the cells. And then when we look at the actual clonotypes called, we see that 64 of these are unique alpha-beta sequences, which means that we are really getting unbiased clonotype amplification. And then this is the sort of data we would expect to see from a PBMC population. So the second collaboration that I worked on was with a group at the Ben Araya Research Institute in Seattle. And this group were looking at CD4 positive T cells that they'd sorted into 96 well plates. They set up controls as per the user manual. So we have 4 JERCAT control RNA wells and 4 negative control RNA wells in the plate. And then, as I mentioned for the previous 2 sets of data, they established the confident clonotype calling threshold using the negative control wells. So here they just did 1 plate and it was sequenced at a much higher depth. So the number of reads you see in these negative wells are slightly higher, but that's sort of with a concomitant increase in the cell wells. So here our threshold cutoff is 607 reads. And reads, clonotype calls with fewer than 607 reads were omitted from further analysis. So we go through the data and we remove all the clonotype calls with fewer than 607 reads, and then examine the amino acid sequences of the CDR3. because we will get non-functional sequences in our data. So all sequences that contained a stop codon or a frame shift were removed. And after that, I'll show you a plate map here. This is what we see. So this plate shows all of the calls that were above the threshold and were functional. So the first thing I'd like to point out is the positive control wells, which are here on this diagonal. These only contain JurCat alpha and beta sequences, so no other sequences are identified in these wells, so that indicates that there's no barcode crossover. And in the negative controls wells, as I just explained, we see a few hundred reads. We use those to set the threshold, and then calls below that threshold are thrown out. And so in this case, you can see, again, there are 7 wells, coincidentally, where there's no call, and that's because they fall below the threshold. So I'm just bringing this table to the next slide, as I'd like to talk about it in a little bit more detail and sort of discuss the pairing information that we see. So if 4 wells are the negatives and 4 wells are the positives, there's a total of 88 wells where we can obtain cell information. We identified at least one TCR sequence in 81 of these wells, so that's 92% of the plate. And the pair was identified in 64 wells. However, 7 of these wells contain 2 functional alpha sequences. So if we look at wells where we just see a single alpha and a single beta sequence, that's 65% of the plate, or 57 wells. So with that, I'll hand back to Ankita for the summary.
Thank you, Sarah, for presenting the data on the TCR, single-cell TCR kit. I'm just briefly going to summarize the presentation for today. So what we've presented to you is data on our newly launched Smarter Human Single-Cell TCR AB profiling kit. With this kit, one can obtain full-length VDJ sequence as well as paired information for TCR alpha and beta sequences. We generate Illumina-ready sequencing libraries, and we can also look at diversity data for alpha and beta, as well as this kit allows for pooling of 96 cells into 12 sequencing libraries, enabling for ease of use. This kit is available in 96 and 480 reaction sizes. And to learn more about our other products, as well as read the literature on our kits, please visit our NGS resource portal to look at tech notes, webinars, posters, etc. And with that, I'd like to close the webinar for today. Thank you all for attending, and for the questions that have come in, we will get back to you with an email in the near future. Thanks all for attending.